Hello, everyone. Welcome to Biology 109 Lab. Today we'll look at, which lab is it? Let's see. Oh yeah, lab number 5 on cellular respiration, whatever that is. So let's get moving. What I'm going to do is turn on the document camera so that you can see what I'm doing there. And that's the wrong button. Whoops. Okay, let's go back here. We're going to go here and it's this button. Okay, right there. It should zoom in and focus here any second now. Maybe. Okay. There we go. All right. So here's the lab number 5 on cellular respiration. We head on over here. We'll take a look. Let's just look at a few things here, and then we're going to have a discussion about this whole thing. Not sure if you understood the introduction, so I'm going to give you a little longer introduction to it that I find very helpful. Something I do in my lecture class too for this material, because it's kind of weird. It's kind of weird stuff. Okay, this is about energy. Plain and simple. It's about energy. That's it. It's energy. All right, so here's what we're going to do. Get rid of this. And we're going to go have a little chat about energy. And I'm going to illustrate things on just a plain old piece of white paper as we go along here. First off, everything needs energy to stay alive. All right, that's kind of a given right there. All right, so in energy, we need other things too, right? We need food. We need to have raw materials to build bodies up, all that good stuff. All right, so here, we're going to start off with some molecules, all right? So this can be really complicated. I'm going to try to break it down, make it pretty easy here. All right, there's a molecule, all right? I don't care what it is. It's just a molecule of something, And here's another one. Here we go. So I have two molecules, and I hope you don't mind. I'm going to abbreviate them with the letter M for this. Now, when we have two molecules, and they're gonna combine or bond together to form a bigger molecule. We're gonna do this. I'm gonna have M to M, so two molecules. That line is a bond. You can think of it, they're holding hands or they're somehow attached. All right. So how does that bond occur? How do we get that bond? How do you get from here, two molecules, to putting a bond between them? Well, it requires energy to put that there. You need an energy input. You need an energy input. So you need energy into the system. You need to add energy to those two molecules to make them bond. It requires energy to make a bond. Requires energy to make a bond. All right. So there we go. But what if we need to break a bond? What if we have this and that's too big? We need to break it down into its constituent parts like this. You take it and turn it back into this. All right, so we're going to do this process. We're going to do this process. We're going to break that bond. Well, when you break a bond, it releases energy. So I'm going to do this. We're going to break it. We're going to release that energy. There it is. There's our energy that's been released. Energy has been released right there. Okay. So when you want to make a bond, it requires energy. When you want to break a bond, it releases energy, and then we can use it. Now, there is a saying in physics and chemistry, of course, that energy can neither be created nor destroyed, only changed in form. Energy, oh, it's sloppy, isn't it? Energy, I think I'm using too thick of a pen, huh? Neither be created nor destroyed. I'm just going to continue because that's how important this is. Only changed in form. All right. This is called the first law of thermodynamics. First law of thermodynamics. Thermodynamics. all right, which is known as the conservation of energy. All right. Not all forms of energy are usable. Not all forms of energy are usable. Energy can be dissipated. All right. It's not really lost, but it's dissipated, lowered in intensity by dissipating it, spreading it out. And energy can spread out. It can be dissipated as heat or sound, for example. Sound, all right, as heat or sound. That's important to remember. Not all energy can be used and harnessed. Some of it's just spread out as heat or sound. That energy is still there, but it's spread out. All right, moving on. Let's consider food, because now we're getting closer to what the lab is really about. Let's consider food. Food. All right. Yeah, all living things need to eat. All living things need to eat. Now, to obtain that food, they either make the food, okay, make the food internally, all right, through a process called photosynthesis. Long word to spell, huh? Okay, which would be some bacteria can do this. Some bacteria, some protista, all right, and of course, all plants. All plants can do this. All right, so we have make food, or they can absorb food right through the body wall, absorb food. So here we have bacteria, right? All bacteria can do that. Some protista, ISTA, some protista, and fungi. There we go. And the last is they can ingest their food. All right, that's an easy one. Some protista, all animals. There we go. All right, these are the three ways that we can get food. Living things can get food. But before it's used, food must be broken down in size because food is really, really big. Before it can be used, it's got to be broken down into smaller parts. So you start off with our food. I'll switch my colors here. We will go with, for our food, we're going to do like this. So here's a food molecule, but made of smaller molecules, all right? So molecules can be big or smaller. All right, here we go. So there's our food. We got to break it down. In the process of breaking down that food, of doing this, of taking this, breaking that bond, making smaller particles of food, we're releasing that energy. So not only are we making the particles of food smaller, we're releasing energy in the process. Well, that energy is neither created nor destroyed, changed in form. That's what's going to happen. It's going to change here. It's going to go somewhere else, all right, with this release of energy. Now, food is not your true energy source. I know you think it is, but it's really not. Food starts this process. When breaking down that food or digesting the food, it releases energy. That energy is then used to build up your true energy source, the real chemical that living things use for their energy source. All right. The food is not really the energy source. The chemical that's made thanks to that energy from food is what we're really talking about. It's what we're really going for. All right. So what we're going to do is change our color again as I can. That chemical, we're going to represent it with a P because we'll define that in a bit. But right now, that chemical actually has a couple of phosphorus molecules in it. And so it needs to bond. We need to put that P back on there. So what we do is we take this energy from food and it goes dink, 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 dink over here and builds that bond back up. All right. That energy from food is used to bond together these two phosphorus molecules. That's an important thing. All right. Well, okay, that's wonderful. That's stored energy. But what about when you need to use that energy? Well, you probably figured it out already. Really easy. This bond needs to break. So when you need that energy, the bond needs to break. When this bond breaks, there we go, breaks, maybe I should rewrite it over here. So when that breaks, there we go. There's the bond. The bond breaks. It's going to release energy. All right. That energy is then used to wherever it's needed. So I'm going to go over here, make a little arrow. That energy goes to where it's needed to do things like move, to think, and to grow. These are the biggies. There we go. That energy is used there. So it's used to make muscles move. It's used to make the nerves in your brain work. It's used to help make your cells put together other chemicals so you can grow in size and replace cells. Okay. Moving on. Pages are stuck together. Bear with me. There we go. Okay. So basically, this phosphorus, this P to P, this phosphorus thing here, blink, is a storage molecule. It's storage. It's an energy storage molecule. The energy is stored in that bond. All right, that's a pretty valuable chemical. Now that chemical actually looks a little more like this. I'm going to draw more of it. I'm not going to draw all of the chemical because we don't really care in our class what the rest of it looks like. We'll just look at the really cool part of it. So I'm going to use the letter A to indicate the name of the rest of the molecule, but we don't care. All right. It starts with an A. It's called adenosine. I'm going to write it down. We don't need to go in the details of it, but it's a bigger molecule. It's bigger than this, but I'm going to use the letter A, and it's got a bond here to another molecule here, and that molecule is a phosphorus and a phosphorus and a phosphorus. All right. And we have bonds. Wink, wink. There we go. High energy bonds. Now notice that these phosphorus are, well, there's 3 of them. There's 3 phosphorus. All right. So it's adenosine plus some phosphorus in an arrangement that includes oxygen and hydrogen, some other stuff. So we're going to change the ending of that word. All right. We're going to have adenosine plus 3 phosphorus plus oxygen and hydrogen. And that molecule is called adenosine 1,2,3 triphosphate. Tri means 3. So it's adenosine triphosphate. There we go, 3 phosphorus. And this is how we're going to represent it. In fact, I like to draw a circle around the P's so I can find them really fast. And that's where the action is. These bonds are high energy bonds. So really this is energy storage. It's a really important chemical. All living things use it. Now, phosphorus is a very high energy chemical. The bonds between phosphorus molecules, wow, so much energy stored there. So much. Now that means when you break that bond, it releases a ton of energy, just a ton of energy, lots and lots and lots of E. All right. So here we go. We're going to take our phosphorus to phosphorus here. We're going to break that bond and it's going to release lots of energy. All right. Let's remember that. Lots of energy. There we go. Okay. What's going to happen there? All right. So after that bond is broken, though, let's go back to our adenosine 1, 2, 3, put our circles here. All right, so we're going to break one of the bonds here. We're going to snap that off. We're going to release that energy so we could use it. What you end up with is adenosine, because nothing happens to that. Nothing happens to the first phosphorus. Nothing happens to the second phosphorus. It's, however, lost the third one. So now we have adenosine 1,2, or adenosine diphosphate, di is 2, which is for short ADP, adenosine diphosphate. I think I forgot, adenosine triphosphate is abbreviated ATP. There's ADP plus a spare phosphorus just floating around. Well, we're not going to waste this because this is a very valuable chemical in living systems. We're not going to waste any of this stuff. So what we'll do instead of wasting it is we'll recycle it and we'll make ATP all over again. And the way we recycle it is, well, we put that phosphorus back on. We're going to have to make a bond, though. What do you need to make a bond? From a few minutes ago, you need energy. All right, you need some energy to make that bond. Where do we get the energy from to put that phosphorus back on? You get that energy from food. There we go. That's why you eat. You get that energy, enough energy to put that phosphorus back on. Okay. Oh, I'm running out of paper. So there we go. This is informal, so just relax, everybody. I'm not going to edit this thing. Okay, there we go. We get it from food. Moving on to the next page. So now we look at food. Oh, I don't know. Let's do it like this. We'll say food is F. There's our food. This is a molecule of food, couple of molecules of food bonded together. I'm going to make our bond in red because it's energy. All right, we're going to take the energy from breaking down food to give us this. Okay, we're going to break that. It's going to release energy. We have smaller molecules of food. These are what can go into the cell for other purposes. That energy then is going to be used somewhere else to regenerate that ATP. There we go. So I guess I'm using blue. So it's going to do this. So here's our A. Well, I should use red on that, shouldn't I? There we go. P, our P, and our last P. There we go. We're going to do this. We're going to take that energy in the process of doing this, migrate it on over, and it's going to be used to build that back up and regenerate ATP. All right, so here's what the system looks like. The system looks like this. You start off with ATP. You break a phosphorus off, release energy. Here we go. That creates ADP plus a phosphorus just floating there, and that had released some energy. All right, we're going to regenerate this. We're going to put that phosphorus back on and remake ATP, but we need energy from somewhere. So we need energy. So we need to add energy to this, add energy to it. Where does that energy come from? It comes from breaking down food. I can't say it enough. You really can't. It comes from breaking down food. So once we break down food, we release enough energy. That's the energy used to put the phosphorus back on to make ATP. Life is this process. This is the whole thing. ATP releases energy, have a spare phosphorus, get the energy from food to put the phosphorus back on to make ATP. You're constantly doing this, ATP, ADP, ADP, and this whole thing. If this process ever stops, it's over. It's totally over. So always we need to make ATP, whether you're sleeping, awake, working out, running, doing anything, sitting, thinking, napping, taking a class, you constantly need to make ATP. If anything gets in the way of that, it's all over. Okay. So what do I have here? Just double checking I got everything here. Go here, go here. Okay. This whole process of breaking down food and using the energy to recycle ATP is called cellular respiration. All right, that's the name, cellular respiration. I know it sounds a lot like respiration, which is also breathing, but it's not the same thing. Okay, cellular means it happens in the cells. Respiration itself is an energy-making process. So respiration, energy-making process. Breathing, which is also called respiration, really better called respiring, is not the same thing. All right, so when we're talking about this process, you've got to use both terms together, cellular respiration. All right, that's the topic of our lab. All right, now, Professor Smith, who wrote this lab, also talks about a couple of other terms. I'm just going to throw them out there for you in a way to remember them pretty easily. He said if a process breaks the chemical to release energy, it's a catabolic process, or we say it's catabolism, C-A-T-A-B-O-L-I-S-M, or it's catabolic. And the way I always remember it is, I think of a nice kitty cat. All right, nice kitty cat. And cats like to tear things up. They like to break them. All right. So catabolism or catabolysis is all breaking things down. All right. Well, there must be a word for building things up, and that's anabolism or anabolysis. That is to build them back up. So it's making, and here's how I like to remember it. I know it's a dumb way to remember it, but it works. And this is Anna makes a cake. Makes a cake. There we go. I know it's dumb, but it's a great mnemonic. It works. Cats tear things down. My friend Anna makes a nice cake. There you go. So building down, breaking down, building things up. There we go. I know it's silly, but it might help you along figuring this out. All righty, moving on. So there are two types of cellular respiration, basically two types. All right, so cellular respiration is of two types. One type requires oxygen, and the other kind does not work with oxygen at all. I'm just going to say without O2, make that short. So the one that requires oxygen is called aerobic. Aerobic. All right, air, oxygen's in air. And then we make this negative by putting the prefix "an" in front of it. And we write the same word, aerobic. All right, not oxygen. So aerobic, anaerobic. If you do aerobic respiration, if you do aerobic exercise, for example, guess what? That means you breathe while you exercise. Wow. I kind of planned on that, right? Kind of funny that they even use that term. There we go. Now, the process of cellular respiration, regardless of which kind you're talking about, starts with one molecule, one molecule, nice R, starts with one molecule of glucose. Glucose's formula, yeah, we got to do a few of these, is C6, so there's 6 atoms of carbon, H12, so it has 12 atoms of hydrogen, and then it has 6 atoms of oxygen, C2H12O6. That's its formula. This too is a storage molecule. It's a storage molecule. We can actually draw a shortcut version of what it really looks like this. That's a multi-sided, it's a polygon. There you go. And every one of these corners is one carbon. So carbon, 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 carbon. When we draw structures, carbon is so common that when we draw structures, we just don't even write the letter C in there. We could if we wanted to. We wrote a C at every one of these here. 1, 2, 3, 4, 5, 6. There they are. And that means attached to these are some hydrogens and oxygens, and we're not concerned about where they go because this is not a chemistry class. Okay. So this molecule can be used to power living things. And it usually is. Other chemicals can be used as well, but this is the number one that is used. Like fats and proteins can also be used. They're very big molecules. They have to be digested a lot, which is a slow process, before they can be used in the process of cellular respiration. Carbohydrates are very fast to digest, very, very fast. They're high energy and they're very fast to digest. So that's what's usually used in the process, but in reality, other chemicals can be used as well. All right. Okay, so let's go on to another thing here. All right, let me cap some of my pens. You just trust me, that's what I'm doing. I want them to dry out. There we go. Okay, there. Now we're ready to go back to the lab itself. So there it is. Now you're ready to read through the way this is written here and understand what's going on. There is the ATP ADP cycle here. There's definition of cellular respiration. I'm going to move on here. There we go. And try to find that. Where's the next page of the lab? Oh, that's lovely. It has migrated. So hang on, everybody. Decided to go somewhere else on my desk. There it is. Oh my gosh, that is amazing. All right, so much paper, huh? Everybody, there we go. There it is. Okay, so cellular respiration. We're using a whole bunch of terms up in here. I don't like you to get caught up on the terms. I'd like you to understand the concepts, though. All right, let's do this a different way. Instead of the way it's presented, what I want to do is give you this. This is a lecture guide that I made, and I really like it. It is posted on Canvas for you, so you can grab a copy of this. You could follow along now, or you can just check it out later. Typically, it's this drawing I use in my tests, hint, hint. So let's go ahead and fill this thing out together because it really helps to pull it together. When you see it like this as a process chart, you get a different pen. Just a regular old pen would be good. Find it in this big old box, so bear with me. Boy, sometimes you just have too many pens. All right, now I can't even see it. I'm here. I'm still here. I just can't find the right pen. I want to use a narrower pen so it won't bleed on the paper so much. Maybe you can read it easier. Okay, let's try that one. All right. So let's go through and start this. So we're going to go and start with one molecule of glucose, 1C6H12O6. That's one molecule. I'm going to put it here. It's going to go through a process that all life on Earth uses. That process is called glycolysis. Glycolysis. To lyse is to break something, like the product called Lysol, which kills bacteria by breaking their cell membrane mostly. So it lyses them. Glyco means sugar. So this is going to be the breaking or destruction of sugar into its component parts. Now you know what it means. All right, so all life on Earth does this. Now here's the thing. This process needs a little bit of energy to get going, just like you need some energy to get going in the morning. If you're completely wiped out, it's hard to get up, isn't it? So you need a little bit of energy. We're going to spend 2 units of ATP to get this process started. And it's a bunch of steps. We don't care about the steps. All right, what's going to happen through this process is that one molecule is going to be broken down into 2 molecules of something called pyruvate. Pyruvate. In fact, it's broken down into 2 molecules of this. I'm just going to write a 2 here, 2 molecules of pyruvate. But in this process, it's going to produce 4 ATP, but how much did it cost us to get it started? 2. So we've got to go 4 minus 2 equals 2 ATP. That is our profit. That's our profit. So our net gain, for those of you who are majoring in business, our net gain, what the profit is 2 ATP, right? Cost us 2, we made 4, We got to substitute. We've got to subtract out what it cost us to get going, and we get our net gain of 2. All right. All living things do this process, right to here, to pyruvate, boop, done. They do continue, depends on what's going on. Now, if oxygen is available, then this is the way it goes, right here. Nearly all living things do this. I'm going to write that in. Nearly all living things are going to do this. Even if they do this process, nearly all of them also do this one. It enters another process that's got 3 different names called the citric acid cycle, but it's also called the Krebs cycle, but it's also called the tricarboxylic acid cycle. I'm not going to spell that out, right? It's got 3 names, but it's all the same process. In this process of a lot of complicated things going on, this also produces 2 ATP, all right? 2 ATP. There we go. Now, this is only if oxygen's available. Then the remaining products, we're not going to name, doesn't matter, they enter another process called the electron transport. I've heard it called electron transport chain, or electron transport system. Do I care which one you use? Not at all. And this one makes a lot of ATP. It makes 32 ATP. Wow, that's pretty efficient, huh? Okay, and then there's some byproducts. The byproducts are carbon dioxide plus some water. Kind of weird. Do you ever have extra carbon dioxide? Do you ever breathe out carbon dioxide? Do you have any water in your breath? All right, these are waste products. And then what's the total ATP that we have made here? We've got 2 plus 2 is 4. 4 plus 32 is 36 net gain ATP, excuse me, from every single molecule of glucose. There it is, the glucose. So going through glycolysis, citric acid cycle, electron transport chain, we produce 36 ATP for every 1 molecule of sugar. That is a pretty efficient process. It's a really detailed process. Okay, that's if O2 is available. What if it's not? So if there's no O2 is available, then what happens depends on what living thing we're talking about. So let's look at yeast and many bacteria, not all of them, but many bacteria do this. They go through a process called alcohol fermentation. Alcohol fermentation. There we go. Alcohol fermentation. All right. Which produces a chemical called ethanol and carbon dioxide. All right. Now, what happens here is no more ATP is produced. None. So what was produced way up here in glycolysis? It was 2. So the total here, the total ATP is simply 2 ATP. That's all. Wasn't even made in this process. It was only made in glycolysis. That's really inefficient, but that's all they need. These are mostly low energy things. Wait a minute, ethanol. Have I heard that before? Yeah, you might have heard of it before. It's the stuff that is used in, there we go, in making alcoholic drinks like beer. All right, beer, wine, champagne, all that good stuff. These chemicals, these ethanols, are made from all kinds of sugar. It doesn't matter what you use. For example, in Japan, they have perfected a formula to make ethanol in a drink called sake that's made from rice. Rice contains lots of sugar. You ferment sugar right here, just deprive it of oxygen, and it ferments and it makes ethanol and CO2. The bubbles in liquor, like when you open up a beer, you get a lot of bubbles, or if you shake it, you weren't very smart, or if you uncork champagne, it spills all over the place. That's CO2 bubbles from this. Basically, CO2 from this process, yeah, these are yeast farts. That's exactly what they are. All right. Well, going back up here, what if you have animals like mammals? I'm going to put mammals here. This happens in pretty much all animals, but I'm just going to use mammals as our example because we're a mammal and this happens to us. If you don't get enough oxygen, you can go through a different process. So let's say that, I don't know, bees are chasing you and you haven't run in quite a while, but now you're running. You go running, you need lots of ATP to run, but you don't have very much ATP available. So your body goes into this process. There's not enough oxygen getting to your cells. In fact, the whole idea of being in shape, it means that your system is efficient at bringing oxygen to your cells. That's what it means. All right. If your system is, if you're out of shape, your system's not used to bringing O2, or you're not breathing enough, or you need way more oxygen than you possibly could breathe, well, you're going to go under this process right here. You're going to go under a process called lactic acid fermentation. Lactic acid fermentation. There we go. Lactic acid fermentation produces lactic acid. There it is, lactic acid. Well, no ATP is produced from this. You still have your total ATP from glycolysis at 2 ATP. So when you produce pyruvate, it builds up. If there's no oxygen, it's just going to build up and build up and build up. And so a lot of things have evolved this great mechanism to deal with lactic acid, just to move it around. It actually goes to a different area of the body and just kind of sits there. And this is the process that made the ATP. It would be great if pyruvate could be further changed to produce more ATP, but nothing alive on this planet has evolved the ability to do that. We just can't. It just ends at pyruvate, and that's it. If there's not any oxygen, it can't go on to these processes. So that pyruvate, which keeps building up, just sits around and it goes into this process and is stored in your muscles. And I know you've experienced this. Lactic acid, being low in pH, burns when it hits your muscles. Very uncomfortable. So if you do a burst of exercise that you've not done before or haven't done in a long time, you're out of shape, you're going to build up lactic acid, and that's going to cause pain in your muscles later on, right? The next day, oh my gosh, your muscles hurt so much. And it takes a while for that to go away. The reason is the lactic acid stored in your muscles where it causes pain, and slowly after you're done with this event, slowly it migrates to your liver where it's converted back to pyruvate. All right, it's converted back to pyruvate. And this happens. Does this. It's converted, so it goes here. It's going to go right here and here. And it's converted back to pyruvate. Why couldn't it do it in the first place? I don't know. Goes back to pyruvate, and now the event is over, so you can breathe enough to get enough oxygen. And now that pyruvate goes here and enters this process and makes lots more ATP. So really, the molecule is not completely wasted. It just doesn't work when you need it the most. It would be great if this could be converted back to pyruvate and go through here, but this won't proceed if there's no oxygen. It won't go any farther. So you're kind of stuck. Pyruvate builds up, and that's why it just gets shunted off and stored in the muscles, and does that right there. Okay, so which is the most efficient process? Well, clearly aerobic cellular respiration right here to make 36 ATP per single molecule of glucose. That's a great way to do this. All right, now you know about alcohol fermentation, lactic acid fermentation, and then normal aerobic respiration. All right, moving on. Okay, so back to the lab here. Okay, so it talks about the different chemicals. I'm not going to read this to you. I'm really not. I just wanted you to understand this. Now you understand what's going on here. They've written out the formulas here. I don't care if you know the formulas. I really don't. Part of the lab later on, the post lab does ask you to rewrite some of these. Our first experiment is about anaerobic respiration, specifically fermentation. And so it goes through the process I just did right now. Depends on what critter you're looking at, whether it's alcoholic fermentation or lactic acid fermentation, and that's what's going on here. Let's go on to our next page, page 59. There we go. All right, here's 59. So 59 talks about this really fun experiment. It really is a really fun, super easy and really fun. What we do is we get some small test tubes and some bigger ones. We put in some liquid that contains, in some of them, in one tube, glucose, which we just talked about is very useful. Another tube has maltose. Now here's glucose. It's really a six-sided molecule. Here it is, glucose. Maltose is actually two molecules of glucose holding hands. Sorry, I misdrew that bond. The bond is in a V shape like this. It's actually kind of cool looking. That is maltose right there. It's two molecules. This doesn't do a thing while it's together. It's got to be digested to be useful at all. And then distilled water, which looks like this. That's H2O with no sugar at all. All right. What we're going to do is see if yeast will feed on glucose and go through that fermentation process. Let's see if they'll feed on maltose and go through a fermentation process. Let's see if they'll feed on distilled water and go through the fermentation process. As I said a bit ago, one of the products of the fermentation process is carbon dioxide bubbles. So we're going to look to see if carbon dioxide bubbles are produced here, because that's how we see if yeast are doing their thing, if they're breathing and doing respiration. All right, so we fill up a test tube. Here's the little test tube. We fill it up with the liquid. Remember, we have 3 small and 3 big. We put that little test tube, we don't balance it on our finger. It's a funny drawing. We hold it and we put this big test tube upside down over it. Once that big test tube's all the way down like this, and we flip the whole thing over rather quickly, some of the fluid's going to spill out. That's what they showed in the drawing. Some of it spills out. That's okay. And then we look at the height of this right here. How much height here, right here? All right, that's air. So we're going to measure that. If we've done this right, there's no air right there. Now there might be a little bit when we do the lab, but there's no air right there. So as the yeast undergo fermentation, they're going to release CO2, and those are going to well up as bubbles, which are going to end up here, CO2. All right. So then there's going to be this airspace here, right? We're hoping not to start with any airspace, but as this goes through, the CO2 is going to make bubbles here. Those bubbles are going to push the liquid. It's going to push the liquid, and the liquid's going to go here and out, and it's going to end up outside of the small tube, but stay inside of the big tube. So we'll see that as displacement. All right. To figure out a measure of respiration of the yeast, we will measure this. We'll measure the size of the CO2 headspace right in through here. All right, that's what we're looking for. All right, so we're going to do it for all of these here. There's actually a formula. They kind of walk you through it here. I wrote the formula here. So you're gonna start off by figuring out how much headspace is here at the beginning. Is there any headspace here? Is there any air right there? All right. And then, so you record how high that is, if there's any air at the beginning, right here, that much. And then that would be your initial size of air. And then once the experiment's done, you measure this all the way down, the whole headspace. And so you take that final headspace size and you subtract the initial from it. So it's final minus initial, and that's what I wrote down here. Your final air bubble minus your initial air bubble quantity. So you solve that part of the formula, and then you divide that over 60, And that gives you the carbon dioxide rate of production, or better said, the rate of carbon dioxide production, which would be a number per millimeters. That's how much this moved per hour. That's why we divide by 60. Okay. That's how that works. So what we do, because we're not doing this, we're not in person, we are giving you the data. We ran the experiment and we ran the data. So you need to jot this stuff down right here. So our start time here, we're going to say is 9. Our end time is 10. Our initial height is 2. Our final height's 15. All right. Our rate of production, we ran it through the formula, is 0.22 for this one, 0.12 for this one, 0.00 for water, which makes sense because it can't feed on water. There's no sugar in it, so it's not going to produce CO2. Glucose, it's going to produce the most because that's the perfect thing to use in cellular respiration. It's going to use glucose primarily. Maltose, it can't use as much. This tiny, tiny number shows that some maltose was used, but in reality, what happened was this maltose did this on its own. Here's the bond, right? It's bonded together. That bond randomly broke for whatever reason. It randomly broke during the experiment, and that released this 2 glucose molecules that are just floating around, and then the yeast fed on these. That's why we got a number here at all. If this was pure maltose that never broke down, this would be a 0 as well, because they cannot use maltose as it is. It's got to be digested, and we're not letting them do it. So these randomly broke, or maybe those yeast were able to break down some of the maltose to glucose, and that's what they fed on. Okay, so you need to read the data and answer these questions. I'm not going to answer them for you, but you need to know these. Down here, we want you to make a bar graph. Make a bar graph. So we want to go glucose, maltose, and deionized water. All right, that's what goes there. And then your fermentation rates are going to go here. So let me go back and look. We have 0.22 is our rate of production. So what if we go 0.5, 1, 1.5, 2. Oh, I've got the decimal points in the funny place. All right. So 2.5, let's just assume I got the decimal points correct. And then we have 3. All right, good. All right. So again, I got decimal points wrong. So this should be 0.05, 0.10, 0.15, 0.20, 0.25, 0.3 in there. So if we have 0.22, 0.22 is going to be right about here somewhere, right around here somewhere. So I want you to do this. I want you to make a bar graph. Use about 4 of these squares and do it like this. There we go. So glucose, there's the rate. You should stretch yours out a little bit more. I just did this real quickly. Okay, maltose is 0.12. So here's 0.1 is the bold line right here. So 0.12 is just slightly above that. So we're going to go here. There it is. Okay, there's maltose. And DI water was 0, which means we're just right down here. This is 0 right there at the bottom. So you can see clearly in a bar graph, glucose was used the most, maltose a little bit, DI water not at all. In fact, I made that too thick. All right, that's how we want you to do that. Make a bar graph of bar graphs out of all of those. Moving on. Second experiment. Aerobic respiration. Well, in this lab, we use these cute little lab mice and we use some fish. We don't kill them. We don't even hurt them. All right, so don't worry about it. We're quite experienced at doing this. All right, so in the process of aerobic cellular respiration, something different's going to happen. All right, an animal's metabolic rate will change with body temperature. All right, so we're going to test that idea. Now, some living things, their body temperature changes with the outside temperature. They're said to be ectothermic. Ecto. Now, ecto means outside. Outside. Things that are ectothermic, like fish, like amphibians, and insects, they don't generate a body temperature. They rely on the outside temperature, and their body temperature follows whatever the outside temperature is. So when it's cold, they're cold. When it's hot, they're hot. They can't make any changes. All right, those are ectotherms. So fish, let's do here, fish and amphibians, like frogs, toads, and salamanders, and of course, insects and their relatives are all ectotherms. Their temperature is going to vary with the outside environment. Well, we're going to hypothesize that their respiratory rate is also going to change with the outside environment. All right, so we're going to test that idea. We're going to use fish, and again, we're not going to hurt them. All right, we're just going to give you the data from doing this. Let's go on to our next page here. So now we're going to start getting into some numbers. There we go. Okay, so we're going to gather some numbers, and here are the numbers. So you need to jot these down in your lab numbers. So we ran it 3 times at 5 degrees Celsius. That's pretty cold. And it had 22 beats a minute. This one had 23 beats a minute. We had 19 beats. All right. We did the average. Now, in case you don't know how to do the average, I put the formula up here. The average that we're talking about is also called the mean, or X bar. It's X with a line over it. And it's the sum of the n's over the n, whatever that means. Well, all of these observations, 1, 2, 3, are n. Each of these are n. That's an n, that's an n, that's an n. So what we do is we do a summary of the n's right here. So we go 22 plus 23 plus 19. There they are. We get 64. And then we divide it by the number of observations. 1, 2, 3. 3 observations. 64 divided by 3 is 21.33 repeating. And that's the number we put here. We use the average or mean quite often in this class, so you need to know this formula. Otherwise, I would not ask you to know formulas, but you need to know how to calculate the mean. It's pretty straightforward. Okay, so then we have room temperature, which is about 20 degrees Celsius. That's about 73 degrees Fahrenheit, right around there. So the beats were more. Look at that. Here's few. Here's more, 26, 29, 25, the mean being 26.7. And then warmer, 35 degrees Celsius. No, we're not boiling the fish. Boiling would be 100. We're not going to boil our fishy. So we had 34 beats, 36, 42. The average is 37.3. You can see right there, fewer beats cold, more beats room temperature, more beats warm temperatures in our fishy. Okay, we're going to graph that, but we're not going to do it right now. We're going to do it later. The graph is going to look something like this. Temperature on the x-axis, because we control it. So dependent variables, like things that we control, go down here, and the response in your organism is on the y-axis, or the dependent variable. All right, independent variable, dependent variable. This is going to be the respiratory rate. We're going to use the averages. So this would be 21.3, 26.7, 37.3. You can see right there it goes up with temperature. All right, our apparatus looks like this. We're going to do a mouse. I'm sorry, I should have introduced Mousy. We're going to use a Mousy. Again, we're not going to hurt him. We're not going to kill him. Don't worry about it. There's our Mousy. He goes in a little jar, and we control how much oxygen goes in there. We use a chemical here that absorbs carbon dioxide. It pulls the CO2 out, so he's not going to suffocate. As he breathes out, he produces carbon dioxide. This stuff's going to absorb the carbon dioxide. He's going to try to breathe in more air, and that air actually comes from this interesting apparatus here. Because I don't have it to show you, we're just going to move on. Okay, so here, this is just a whole area on page 63 that talks about weighing the chamber and the mouse and figuring out what the mouse weighs. Well, the weight of mouse is 15 grams. Just trust me on that. That's what the one we did for the test experiment here weighed 15 grams. All right. And then we have to use the apparatus. Without actually having an apparatus, this stuff won't make sense to you. So let's just move on down here. We're going to fill a syringe. No, we're not hurting the mouse with a syringe. The syringe contains air. It contains air. It's going to contain 2 milliliters of air. All right. That's going to be the oxygen. And later on in the calculations, that's a variable called oxygen consumed. That's where you get it from right there. It's why I started. 2 mils of air are going to be important in the calculation pretty soon. We record the time it takes for fluid levels to equal out, which means how long did it take it to use up 2 mils of air? That's what that means. All right. Down here, we're going to repeat it a number of times because we always repeat stuff. We're going to put the numbers into table 5.4, which is on the next page. This page, this table down here is from this page. This is where the times go. That's table 5.3. I pointed out because it's very easy to put the wrong numbers in the wrong table. Very easy to do. So table 5.3, your table 5.3 needs to look like this. So in the one we measured, it was 5.13 seconds, 4.75, 4.59. We did the mean of that. It's 4.82. That was at 20 degrees Celsius. We don't do 3 measurements for the mouse. We only do 2. So then we do a cold temperature, and we got 0.74, 0.85, and 0.79, and the average is 0.79. Those are the numbers that need to go in your table in your lab, so that you understand this. Okay, moving on. Now, page 64 here. All right. So here's table 5.4 for our mousey. And we put in the weight, and the weight didn't change. So it's 15 grams in both. The average time we took from the prior page, 4.82 and 0.793. Respiration rate is what we calculated down here. The way this formula is written is kind of confusing. So what I did is I added parentheses here. No, it looks kind of sloppy. I made it sloppy on purpose so you could see it. Put parentheses around these numbers. So when you enter this into a calculator app, you do it like this. You start off by clearing your calculator app. So go all clear first. And I always hit it twice at least. All clear, clears everything out. And then I'm going to work on the bottom of the equation first. I'm going to go weight of animal in grams, 15, times average time in minutes, right there, 4.82 equals, all right, so I get that. And then I go memory plus in a calculator and calculator app. So I want it to store that number in memory. And then I hit clear, not all clear, just clear. Just hit clear on the calculator to clear out those numbers from the temporary memory. It's still in the stored memory, but it's not in the temporary memory. So then we hit clear in our calculator app, and then we type in the oxygen consumed mil. Well, that's 2 mils. All of our experiments worked with 2 mils. So we put in a 2, and then we go divide by memory recall, right? That's the MR, the memory recall button on a calculator or calculator app. And then we hit equals, and that gives us the answer for this top one, which is 0.02766. And that's what goes into this table right there, 0.02766. And then you multiply that by 10 and go ahead and round it off a little bit and put it in this table. I know we didn't give you a column for that, but it goes right there. We're revising the lab manual. It'll have another column later. Okay, then do it on the bottom here. Do the formula the exact same way. 15 grams and also 2 mils. That didn't change, but these things, this one did change. So plug that in. Follow the exact same way to do it, and you get this number. So now we have our mouse response at room temperature and at cold temperature. We did not do a warm temperature with our mousey. Room temperature is fine. We don't want to warm them up, right? We're very careful with our animals. Okay, now here's the graph that we want to do right down here. We're going to graph the mouse and the fish on the same graph. Now, don't sweat the details on this graph. It's very easy to see it and go, wait a minute. The units aren't the same on the left and the right side. That's true, they're not. We're going to put the fishy on the left side of this. Here's our fishy. So our fish graph is going to be 5 degrees and then up to 20 degrees. All right, it's going to be cool to high and low respiratory rate to high respiratory rate. So when it was cool or cold, I'm going to say cold, Our fish's respiratory rate, and I'm going to put an F right there. Our fish's respiratory rate was low. Let's see if I have that data here. So our fishy, when it was cold, was 0.79. So I'm going to say 0.79. Don't worry if the units don't match left and right. These axes use different units. We're just going to look at a trend, and our graph is going to look like this. So for our fish, our cold rate is 0.79. No, it's not. I'm looking at the mouse, aren't I? Whoops. All right, where's our fishy? I didn't go back far enough. Sorry. There we go. There we go. Okay. So our doink, not so good. So our fishy was 21.3 here. And then middle temperature, which we didn't label, it's okay, went up to, when it was room temperature, went up to 26.7 for our fishy, 26.7. And then when it was warmer, our fishy, yeah, I got the nice fishy there, 37.3. So it went up. Now look at the trend, 21.3, 26.7, 37.3, it went up. And you can draw a line between those so that you can see it better. Now we need to look at our mousey, and our mouse, let's use a different color. We use let's see, oh, it's already red. Yeah, we use blue. Our mouse here, we only have two values, and they're on the same page. They're right up here. So for our mouse, which will be on this side, our mouse at room temperature was 0.28, and at cold temperature was 1.68. All right. So putting our mouse on here, we're going to find 0.28, which is, I don't know, it's down here. It's low, isn't it? There's our mouse right there. Our mouse. So here at warmer temperatures, we have 0.28, but at a cold temperature, because remember, this is cold over here. At a cold temperature, our mouse, let me label this 0.28. Our mouse was 1.68. All right, and then we can draw a line between them there, showing that our mouse and our fish had opposite reactions to the temperatures. So let's go through and make sure you understand this, because this graph is on the practical. All right, so cold temperature, our fish, which is an ectotherm, which is the same temperature as its outside environment, or pretty much. As it gets warmer, its respiratory rate goes up. But our mouse that controls its respiration rate, when it's cold, it actually increases its respiration rate in order to generate more ATP and burn that ATP, break molecules apart, break ATP apart to form ADP, which releases energy, but also some of that energy is dissipated as heat, which makes the body warmer. As it gets warmer, the mouse doesn't need to generate more body heat, so it doesn't. So it becomes really efficient, and it lowers its respiration rate. This is also its rate of breathing too. So it's going to breathe less actually. So looking the other way around, when it's colder, endotherms like you and the mousey have a higher respiration rate, and that is to generate some excess body heat and keep it warm in the cold, where fish they're just tough. They're so much tougher. They can handle that cold. They don't freeze. They freeze at a much lower temperature than we do, and they can handle it. And their respiratory rate matches the temperature of the water, where in endotherms, like you, me, and the mouse, their respiratory rate is the opposite of the environment right here, okay? And then after doing this, you've got to do the graph first. Then you can answer these questions. Answer these and know these. They're really important. I'm not going to answer them for you. There we go. Moving on. Okay, what's the next page? That was 64. We need to go to 65. Yeehaw, we're getting there. Now we're on to the post lab. All right, so name. Put your name up here, please. All righty. So now you need to answer these questions. What compound is the main energy? What compound is the main energy carry of the human body? How about source? And right, that's going to be ATP. What is this main energy unit produced from in animals? From eating. Write out the alcoholic fermentation equation. That's right in the lab. Not going to do it for you. Write out the aerobic respiration. It's in the lab. Lactic acid fermentation. It's in the lab. Oh, here's a thought question for you. Not going to do it for you. Okay, our second page of the post lab is here. What happens to the metabolic rate of a fish when the water temperature rises? Go back and read the graph. And then when it lowers, go back and read the graph. What happens to the mouse? Read the graph. Do the results verify the definition of ectothermic organisms? And why or why not? Read the graph and the description of that word. What about endothermic? Read the graph and the definition of that word. How are the results different? Read the graph. So I can't say it enough. Understand the graph on page 64 and its questions. And boom, that is the lab. All right, there we go. Thanks for sticking with me. I hope everybody is actually watching this thing and listening. Great, we're done. Let's call it a day.